The big ideas we want to cover today are things like membrane structure and function, including how things pass through cells and the way that membranes can be attached to each other. So, we're going to spend quite a bit of time on this today, which is part of why it was important to understand how phospholipids work before talking about this. We'll then discuss the basics of energy in the cell because we're going to next week focus a lot on cellular respiration, which is essential for most cells to get access to energy so that they can use that energy to perform chemical work. And we'll spend a little more time discussing exactly how enzymes work and what they do for reactions. The first thing that we really want to understand about the cellular membrane is that it is essentially a fluid. We talked about this idea that the membrane is made up of phospholipids and that these phospholipids form the membrane not because they are bound to each other, but because of their hydrophilic and hydrophobic properties. This means that they can move around. They do have some hydrogen bonds between their tails, but those are easy to break. So the entire membrane is essentially a fluid that is lacking rigid bonds. So those hydrophilic, hydrophobic properties are what make that cell membrane stay. Because of this, we often refer to this as a fluid mosaic. So mosaics are images that are made up of multiple smaller images that individually don't mean a lot, but together can form a full picture. And the membrane is like that as well. So most of the membrane is these phospholipids, but on top of that, there are other things attached to the membrane. Because it is not a rigid structure, the membrane is able to bend, flex, and move. And floating within this structure are other objects like proteins and other lipids. Right? So this is a very flexible structure. That's important too because cells can become damaged. And as long as the damage is small, the phospholipids are actually just able to refill the space. They don't need any assistance in doing so. I'm going to mute this. Stop that. Okay. Inside the membrane and also able to move around within it are proteins. All right, so we talked about how this is a mosaic. Mosaics are made up of multiple different things that together make a complete picture. So while most of the membrane is phospholipids, not all of the membrane is. Peppered through the membrane are other materials. For example, there are structural collagen fibers that are on the outside that help support the shape of the cell. There are proteins that are going to be attached to and pass through the membrane that perform different functions. And there are also other fats like cholesterol, which is a type of steroid, that are going to be in the membrane as well. So there are lots of things that are attached to the membrane, and all of these are going to perform slightly different functions. Now, the most important things that we're really going to discuss about this are going to be membrane proteins. So all of these are purple things in the membrane in this image. Much like the proteins we discussed last week, membrane proteins have multiple functions. But because they are attached to the membrane itself, they only perform that function at the membrane. So they do not perform their functions anywhere else. Some examples of protein functions for the membrane are things like attachments. So proteins in the membrane can allow cells to attach to each other, or they can allow things like the cytoskeleton or extracellular proteins to attach to the membrane. So they form as anchors. Receptors are also often part of the cell membrane. So we discussed how proteins can be used to both send and receive signals. Most chemical receptors that are necessary for receiving signals from other cells are going to be located in the cell membrane. There are enzymes that are also embedded in the membrane. This causes them to perform their catalytic function, their ability to cause bonds to break or form at the membrane specifically. Proteins are also used for cell recognition. So, cells in a multicellular organism, like a human being, need to be able to tell which cells belong and which cells do not. So, the cells that both present their ID and the ones that read the ID, those are going to be done by proteins too. And proteins are also necessary for many types of transport. So, because the membrane forms a barrier between the inside of the cell and its outside environment, many things are not going to be able to get inside the cell without help, and proteins can do that. So, you should be aware, much like the proteins previously, that the ones in the membrane also have a wide variety of functions. Those functions are going to happen at the membrane specifically. And so, lots of proteins floating around in there. We can look a little closer at one of these examples. So we're going to look a little bit at this attachment selection of proteins. So these ones here that are necessary for allowing cells to connect to each other. Multicellular organisms need to be able to form tissues and organs and other structures where cells are tightly linked to each other. But the cells that are part of a tissue need to be able to work together in very specific ways. When I talk about junctions, of which there are three tight junctions, anchoring junctions, and gap junctions, I like to talk about them in the context of the intestines. So let's think for a minute about these cells. So this cell here is an intestinal cell. The structure of your intestines looks kind of like this. You have a bunch of cells that are all next to each other. Right, so this is an example of what looking at your small intestines would be like. So in the center here is going to be where you have food. And these cells are going to be important for both getting nutrients out of what you've eaten, but also preventing any of the waste from getting through. And to do that, they need to have all three of these types of junctions. So this is a good tissue to think about that in. The first type of junction is called a tight junction. The purpose of the tight junction is to prevent fluid from being able to move between the cell layers. So, what happens is the fluid that's on the inside of the intestines is not able to pass between the cells. The tight junction will prevent that from happening. This makes sure that undigested food and food waste stay outside of the body in the lumen of the intestines. Without the tight junction, this material would be able to seep between the cells and enter the fluid in the rest of the body. You really don't want that to happen. In fact, when it does happen, say if you have an injury to the bowel, it can cause sepsis. So tight junctions are necessary for keeping separate and to prevent movement between cells. Many cells in the rest of your body don't want to do this. For example, your blood needs to be able to send nutrients to all of the rest of your body. That means that the cells that hold together your bloodstream don't have tight junctions. If your blood pressure gets too high, fluid is easily pushed between the cell layers, and that's part of what causes swelling and edema. Because blood needs to be able to transport nutrients, there is unfortunately that side effect. But for things like your intestines, where it's absolutely essential that doesn't happen, tight junctions can prevent it. The other type of junction, the anchoring junction, behaves a lot like a drywall anchor. So if you've ever had to hang something in a wall, what happens if you just put a nail in drywall and you try to hang something? Have you ever done that without using an anchor before? When you want to hang something in a wall, are you going to just hang it wherever or are you going to try to hang it in a stud? Does that depend on what you're hanging? Yeah, it depends on what you're hanging, right? Let's say that I want to hang something that's really heavy. There's not enough support without an anchor, right? So if you want to hang something really heavy in a really thin piece of wall, and all you do is put a nail through it, then your options are basically that the wall is going to tear and pull out, or the nail itself is going to pull out. So the purpose of a drywall anchor is, of course, to spread the force. So when the drywall anchor is used around a screw or nail, it causes the anchor to spread out. This reduces the force in a specific location and spreads the force across more of the wall. Essentially, that is the same thing that the anchoring junction is doing. Because the membrane is a fluid mosaic, it is not particularly strong. So, what the anchoring junction does is it also has large amounts of tendrils of proteins that it uses to connect to the cytoskeleton of the cell on either side. This means that if the cells are pulled apart, all of the force does not go just to the membrane. There are other anchors now that are helping keep this in place. So, because this membrane is fluid and not a solid structure, if we just tried to connect them with a tiny little connection, it would tear right out. So, the anchoring junction does the same thing as this drywall anchor. It provides strong physical connections. This is the kind of junction that you'll often see in things like muscle or important cell layers. For example, if you've ever experienced a muscle tears occur either when the cells themselves are torn or the anchoring junctions between muscle layers are broken. Because the intestine itself needs to be able to contract in order to push feces along, it also needs anchoring junctions. We want to be able to apply enough pressure to force the digesting food downward, but we don't want the cells to come apart when we do that. So these anchoring junctions provide that strong physical connection that allows us to push things or pull things or carry things without having our cells come apart. The last type of junction is called a gap junction. And this junction behaves kind of like the door in between a suite in a hotel. Now, this isn't as popular as it used to be, but it used to be that if you wanted to go out on a trip and you wanted to stay in a place that wasn't that expensive, you might stay in a motel. And oftentimes, motels have adjacent rooms called suites, and those suites will have a door between them. Has anybody stayed in a place like this where you have a suite, two different rooms with a door between it? That's okay if you haven't. All right, a couple people have. When you stayed in the suite, did you know the people in the other room? You did. And did you keep this door open or closed? Closed at night and probably open in the day. So sometimes open, sometimes closed. Why did you keep it open during the day? Because you knew each other, right? So this is basically what a gap junction does. A gap junction functions like that door. So if you know the person in the next room and you want to be able to go back and forth between the rooms quickly or be able to communicate with each other without knocking and trying to make sure that somebody will unlock the door for you, then you are going to keep the door open. If you don't know the people in the other side or you don't want them to be able to come in, then you're going to keep the door closed. Essentially, what the gap junction does is it creates a direct connection between the cells. This allows information, nutrients, and materials to pass through the cells directly instead of having to move through the membranes twice. So it saves a lot of time. But you only want to do that if you really trust the person who is in that other suite. So gap junctions connect the cytoplasm of cells together. And it makes it fast and easy to transport all different kinds of things like ions and small molecules and certain types of nutrients quickly across the membrane. This is important when cells have to coordinate a behavior with each other. Because the movement in your intestines is always in one direction and it's going to move in waves. So it moves in little waves. In order to coordinate that wave, these cells have gap junctions, which allows them to transport ions. As the ions move from cell to cell, the wave moves too. So this lets that happen really fast without having to deal with moving things through one membrane into this lumen and into the next membrane. So I'd like for you to know the three types of junctions: tight junctions, anchoring junctions, and gap junctions. All of them are junctions that are held together by proteins, but their purposes are different. Tight junctions create a barrier to fluid moving in between cells. This is important for keeping those fluids separate. Anchoring junctions are needed to create strong physical connections between cells, which allows them to apply pressure or survive being pulled without being pulled apart. And gap junctions connect the cytoplasm of cells, making it fast and easy to transport certain types of molecules, like ions, nutrients, or other small molecules. I could either ask you about this type of question by asking you to define the type of junction, or I could ask you to tell me what type of junction I'm describing, or if I wanted to do something, which junction I should use. Are there any questions about proteins in the membrane at this point, or about these types of junctions?
Did you say that all of these junctions are surrounded by protein?
They are proteins.
Okay.
So tight junctions here, each of these little circles is a protein in a mesh network that's creating this junction. The anchoring junction is a really big protein that has these extra like tendrils that are attached to it. And the gap junctions are a channel protein. So they usually are a large protein with a hole in the middle, but they're all proteins. So these are all attachment proteins, so they fall into this category. And they'll be one of these three types. Okay. So now we're going to talk about how things get across the membrane. So we have all of these things set up so that cells can be separate from the environment that they're in. But the reality is that they need to get stuff inside and they also need to move things outside. And there are a bunch of rules about how that works. And they have to do with the chemical nature of the membrane itself. Remember that the cell membrane is a phospholipid bilayer, meaning that it is made up of phospholipids that are orienting themselves in a water type environment, with their heads toward the outsides and the tails to the inside, and that it is a fluid environment and that these can move. Because of the properties of the phospholipid bilayer, the membrane is selectively permeable. That means that some things can move through the membrane without help, but other things cannot. For example, small molecules that are not charged are able to move directly through the membrane. This is because the center of the membrane where the tails are located is nonpolar. So it's easy for them to interact with that. So really small things like oxygen, carbon dioxide, and water can cross the membrane directly. However, larger molecules and molecules that have a charge cannot. In order for large molecules like sugars and amino acids, or ions, atoms with charges, to cross the membrane, a different type of transport is necessary. So, the type of transport that's used for small molecules that are uncharged is usually diffusion. There's a really good explanation of how diffusion works in the videos posted before today's class, but I'll also remind you about how it works in a little bit. So, what are the other options? In simple diffusion, molecules move through the membrane on their own with no assistance. When they do this, they will always do so along the concentration gradient, where they'll go from a high concentration to low concentration. So movement from high to low is with the gradient. This movement requires no help, moves in the direction with the gradient, and because molecules behave this way normally, requires no energy on the part of the cell. Larger molecules cannot pass through the membrane directly, and so they perform something called facilitated diffusion. Facilitated diffusion allows molecules to diffuse through the membrane through transport proteins, which provide large enough openings for the molecule to move. Because this is facilitated diffusion, and the molecules are still moving in the way they wish to move naturally, it will also happen with the gradient. So they will move from high to low. Essentially, the transport protein is just providing a channel, and the molecules are doing all the work. So the cell does not have to expend any energy for this to happen. Let's say the cell wants to move something that isn't in this direction. The cell also has a way to move objects through active transport. Active transport also requires transport proteins. But the transport protein has to perform work in order to move against the gradient. Molecules do not want to move from low to high. So to do this, energy has to be spent. So simple diffusion and facilitated diffusion move with the gradient from high to low. and therefore require no energy. Facilitated diffusion and active transport move large molecules and molecules with charges, so they both require proteins. But unlike facilitated diffusion, because we're moving from low to high against the gradient, active transport requires energy. You should know the difference between these three types of movement, whether they need proteins, whether they move with or against the gradient, and whether they require energy for this to happen. Another way to think about this is to organize them based on what is moved. Remember that in simple diffusion, we can move small molecules that are uncharged. A special type of diffusion called osmosis is the movement of water specifically across the membrane. Facilitated diffusion moves large molecules and ions through special proteins. And active transport can move ions, glucose, amino acids, and other large molecules against the gradient. If we need to move something even bigger, we will use something called endocytosis. Endocytosis allows us to move extremely large objects like whole proteins, other cells, and large food processes through the cell membrane. It requires energy and occurs in two primary ways. Phagocytosis is a type of endocytosis where the cell is essentially moving outward. In this case, the cell detects that there is food and pushes the cell membrane out to create a pocket around the food. Once that happens, it's placed in a food vacuole, and the food vacuole is then brought to a lysosome to digest it. The other type of endocytosis is receptor mediated endocytosis. In this case, instead of the cell pushing outward, the cell detects something that it wishes to bring inside using receptors, and the membrane pulls inward, placing the material also in a vesicle. Remember that vacuoles are just larger vesicles. Both of them are membrane pockets the cell can use to package materials and move them around. If you'd like to see more about how this actually looks in a real cell, remember that there are videos in Module 3. For example, we can see this amoeba use phagocytosis to consume a paramecium. Look how the cell pushes its pseudopods further out to create a pocket. Once the paramecium is inside, the membranes on either side will close, pinch off, and place the paramecium in a food vacuole. Then the paramecium will be digested using lysosomes. So this is an example of endocytosis, a way for cells to bring very large objects, including whole other cells, into themselves. Are there any questions about the types of cell transport? If I were to ask you which ones require energy, or in what direction do they move things, or if I need to move this substance, what should I pick? You should be able to answer. Sure. In receptor mediated endocytosis, the primary difference is that instead of pushing the membrane outward, the membrane pulls in. So it looks kind of like this. Here are receptors, and here are the molecules that they're going to detect. When that happens, the cell says, I want that. I want to bring it inside. So instead of pushing outward, it'll start pulling in. Same idea, but instead of pushing out to create that vacuole, we're pulling in to create the vacuole. Instead of detecting that there's something outside that we want to grab, we're detecting that there's something and kind of sucking it in. Both of these require energy. So to do this, right, in when we talked about the cytoskeleton is that those sets of proteins that create these different structures, right? So to do this, we're building and destroying the cytoskeleton pieces to push the membrane outward. The process of doing that is going to require energy because we're building and breaking bonds to change the membrane shape. So yes, these both require energy. Right. Any other questions about transport? Okay. So next week, we're going to focus a lot on cellular respiration. But before we do, we want to cover a little bit about metabolism and thermodynamics, because that will make understanding respiration a little bit easier. So now we're going to think about energy in general, but also about how energy is something cells need and what that really means. To understand how energy works, we have to go back to the laws of thermodynamics, which is something we probably heard about in elementary and middle school, but probably also don't really remember the definitions of, and that's okay. The first law of thermodynamics simply states that energy cannot be created or destroyed. It's only converted from one form to another. For example, in the fire that we see here, what we are doing is we are converting energy in the form of carbon bonds, because that's what wood is, lots and lots of structural carbon bonds. And when those bonds are broken, that releases energy in the form of light and heat. It's not that energy was created. The energy we're seeing in the fire was in the wood all along. We're just converting it from one form to another. Conversion of energy is something that happens all the time in cells, usually through the making of energy molecules. There's our fire. For example, we can use light energy through photosynthesis to make sugar or glucose. In this case, we are converting the energy in photons into chemical energy. We can also convert different types of molecules into other molecules. For example, we can convert glucose, a monosaccharide, into storage molecules like starch or glycogen, or even fat. We can also create energy molecules like ATP, which is the primary energy currency that's made in the mitochondria through cellular respiration and fermentation. All of these are examples of converting one type of energy molecule or energy type into another. Once we've created energy molecules, the cell can then spend those molecules to perform work. Some examples of this type of energy conversion is the conversion of energy from chemical energy to physiological action. We can convert chemical energy into kinetic energy through movement, or into electrical energy by converting molecules into nerve impulses. All of these are examples of energy conversion. None of them create or destroy energy. They simply change where it is and the form it is currently in. The second law of thermodynamics states that during energy conversions, some energy is lost. Some energy transforms from what we call ordered into disordered. Most usable energy for living organisms is held in the bonds of molecules. This is an ordered form of energy. During energy conversion, when bonds are broken and reformed, some of this energy escapes as heat. This is disordered or unusable energy. This means that energy conversions are not efficient. A classic example of this is a traditional gasoline car. Gasoline cars use chemical fuel, gasoline and oxygen, in order to create a combustible engine. The combustible engine converts chemical energy into kinetic energy. This process is incredibly inefficient, though, and creates a lot of heat, which is why the longer the car runs, the hotter the engine becomes. This is why cooling objects where energy conversion happens is so necessary. Most power sources have built in ways to cool them in order to avoid being damaged by the heat made during conversion. After energy is removed from the molecule, waste molecules are often left over. In the case of the car, the waste products from gasoline and oxygen are carbon dioxide and water. So the combustion output is conversion of chemical energy to kinetic energy. The reactants gasoline and oxygen are converted to carbon dioxide and water, and some of the energy is lost as heat. Cells work essentially the same way. Instead of gasoline, cells use glucose as their primary energy source. They also use oxygen and produce the same major waste products that cars do, carbon dioxide and water. In this case, we're converting one type of chemical energy into another in the form of ATP. This process also produces heat. This is one of the reasons why you have a body temperature that is above the environment. All of the processes that your cells are performing produced waste energy, the side effect of which is your body temp. This is also why all living things need to consume constant sources of order usable energy. They need to get this energy directly from their surroundings. Where do you get your usable energy from every day? Sugars or other types of food. So, all living organisms need a constant supply of ordered energy. And for most of them, that's either going to be sunlight or food. So, if you ever wondered why you have to eat all the time and you're constantly hungry, you can blame thermodynamics. When people talk about metabolism efficiency, i.e., my metabolism is faster or slower, what they really mean is how good the conversion rate is. Individuals that can get more energy out of less food have a better conversion rate and less heat production than individuals that can't. It's not really that your metabolism is faster, it's more efficient. So, why do we have to think about this? Well, we mentioned that most usable energy is in bonds. This is a type of potential energy, stored energy, or energy based on position and mass. Unfortunately, this is something that we often think about when it comes to physics. Most of the time, potential energy, as we think about it, is kinetic energy, stored in the location an object is in. For example, the ball at the top of the hill has high potential energy. At the bottom of the hill, it has low potential energy. This is because as the ball moves from top to bottom, kinetic energy or energy of motion is released. So energy is lost as the ball moves. But to move the ball back up the hill, energy would have to be put in. The potential energy of molecules is a type of chemical energy that is stored and released from chemical bonds during reactions. Some molecules have more potential energy, or energy that could be released, than others. Reactions that release and store energy come in two major varieties. Exergonic reactions release energy. These reactions start with reactants that have high potential energy, or free energy. At the end of the reaction, the products have low potential energy, and the energy difference is released. Some of this energy can be used to perform work, and some of it will be lost as heat. The opposite reaction, the endergonic reaction, requires energy input. In this situation, reactants start as low potential energy, low free energy molecules. Energy is required to be added to the system to create higher energy products. You should understand the difference between an exergonic and endergonic reaction. The relationship between the energy levels of the reactants and products. High energy reactants release energy to produce low energy products in exergonic reactions. Low energy reactants gain energy or have energy added to them to create high energy products in endergonic reactions. These reactions are often coupled together. The energy currency of cells is called E, or adenosine triphosphate, the nucleotide. The parts are adenine, the nitrogenous base, ribose as a pentose sugar, and three phosphates. So the only difference between this and the kind that's part of RNA is the extra phosphate groups. Energy is stored in the molecule in the phosphate bonds. When the bond between the second and third phosphate are broken, energy is released. To break bonds, remember, we use hydrolysis reactions, where water is added. So water is needed for this process. This creates adenosine diphosphate, or ADP. A phosphate, and releases usable energy. ATP is a replenishing energy source. You can think of the molecule kind of like a rechargeable battery. ATP is used by cells to perform work. When this happens, energy is released by breaking the phosphate bond, creating ADP and a phosphate. But ADP and its phosphate can be recharged by adding energy in, recreating ATP. This process can be done over and over again, as long as an energy input source remains available. So your body is constantly looking for energy input, in your case in the form of food, but in plants in the case of photosynthesis light energy, to regenerate ATP for use. This is because ATP can be used to perform many different functions in cells. So we're going to generate ATP from photosynthesis, cellular respiration, and fermentation. ATP can be used to form products through chemical work. In this case, ATP is used to remove the phosphate and create a bond between these two reactants. It can also be used to transport molecules. We talked about how active transport requires energy. Most of the time, this energy is in the form of ATP. Adding energy changes the shape of the protein. Remember that shape is necessary for function. When the protein's shape changes, it's able to push the molecules out of the cell. So this process changes the shape back and forth between the open and closed condition to move molecules, which means it requires constant access to more energy. Mechanical work in cells are also created this way. The motors that allow your muscles to move require energy in the form of ATP, which changes the structure of the motor protein, causing it to move itself along the protein filaments. So almost everything that your cells are going to do, they use ATP stored energy to accomplish. The process of generating ATP is metabolism, and it requires things called metabolic pathways. Metabolic pathways are a series of chemical reactions that make an organic end product. There are two major types of metabolic pathways: linear pathways, where we go from our initial reactants to our final end products with some intermediates. In a linear pathway, there is no going back. We don't need to regenerate anything. But there are also cyclic pathways. Cyclic pathways require us to regenerate a starting material. In a cyclic pathway, the starting materials are the initial reactants. But one of them is also an end product. Unless we regenerate this initial material, the cyclic pathway will stop. So linear pathways do not regenerate materials. They move from A to B to C to D to E in a linear fashion with intermediates. Cyclic pathways have initial reactants that need to be regenerated. So they are both an initial reactant and a final end product. Other components of metabolic pathways include things like enzymes. Enzymes are usually present at every step in which you see an arrow, and their job is to make reactions happen faster. The reaction will happen without the enzyme, but it might take a long time. So enzymes are catalysts that help things go faster. They also involve coenzymes. Coenzymes or cofactors carry small amounts of energy to help enzymes do their job. And they do this by transporting hydrogen ions and electrons. When we go through respiration and photosynthesis, we'll spend a lot of time talking about the specific coenzymes. They're going to be necessary for generating large amounts of ATP. And of course, the metabolic pathway also includes the ATP molecule itself, which can be made during these pathways and can be used during these pathways to convert low energy molecules into high energy molecules. So metabolic pathways can be linear or cyclic, and they will usually also include enzymes, coenzymes, and ATP. In addition to these types of linear and cyclic pathways, there are special types of pathways called electron transport chains. These are chains that move electrons among a series of proteins, enzymes, and coenzymes in a phospholipid bilayer. In order for this to happen, they need a source of electrons for them to carry. And these electrons need to be able to move along a clear path. For the electron to be able to move from the source, it also needs a destination. The movement of the electron between all of these different proteins can then be used to form a substance. Every time the electron moves from one substance to the next, small amounts of energy are released. And this occurs because bonds are breaking every time the electron is transferred. This energy is usually used to transport hydrogen ions, which we will then use to make ATP. We'll go into details about how this works more next chapter. But the electron transport chain is going to be super important. The last thing we want to talk about today is more closely how enzymes actually work. But before we do, how are you guys feeling about metabolic pathways? Feel comfortable with the difference between linear and cyclic? What about types of reactions, like exergonic and endergonic? Okay, practice is always a good thing. So let's think about it a little bit. We're going to talk a little bit about how this works too with our enzymes. Mhm. So I mentioned that enzymes are catalysts. This means that their job is to make reactions happen faster. So how do they do that? Essentially, all reactions have reactants and products, but they also have something called activation energy. Most reactions are not spontaneous. Activation energy is the amount of energy that's needed to cause the reaction to start. In order to get this reactant to move to product, we're going to have to put energy in before we get energy out. Some reactions have extremely high energy barriers. So the time it would take to get enough energy to cause the reaction to start is a very long time. For example, let's say we want to be able to push this rock down a hill, but there's a hump on the hill in the way. We're going to have to push the rock up the hump before we can have it roll down the hill. This is the barrier that's preventing us from being able to do that. The enzyme's job is to reduce the amount of activation energy that is required. Enzymes put strain on the bonds inside reactants. This reduces the energy we need to pay for the reaction to occur and makes it easier and cheaper for that to happen. And this makes it faster. You can think about the enzyme as kind of like a coupon. You want to purchase this object, but this is the price you'll have to pay. It's going to take you less time to save up for the item if you have a coupon that lets you pay less. So you'll get the item sooner. It's the same idea. With the enzyme present, the reaction is going to happen faster and sooner. But it would have happened eventually without it. It's important to remember that enzymes are very specific. For example, this enzyme is an example of sucrase. Enzymes usually only work as catalysts for specific substrates, so they only work with some molecules. In this case, sucrase only interacts with sucrose. The substrate the enzyme works with is its reactants. The reactant has to be able to fit into the pocket or active site that's located on the enzyme. In this case, the substrate or reactant, sucrose, is able to fit in the active site. Once that happens, this puts strain on the bond between the two molecules. We can see this in the structure. It was straight here and it's curved here. Then we can add water and perform a hydrolysis reaction to break the bond, now that it requires less energy to do so. Once that happens, the products of the reaction can be released. It's important to remember that the enzyme is neither a reactant nor a product. If we were to draw this reaction, we would say sucrose has been converted with water to glucose and fructose. Water and sucrose are our reactants, and glucose and fructose are our products. But sucrase is neither a reactant nor a product. At the end of the reaction, it looks just the same as it did before, which makes it infinitely reusable. When looking at a reaction, the enzyme is often written above the arrow. This is where you'll find the catalyst. So enzymes are infinitely reusable. They are neither reactants nor products. They are catalysts. They work by reducing the activation energy required for this to happen. A catalyst is anything that makes something happen faster. So an enzyme is an example of a catalyst, but catalyst can also be a chemical. Like let's say you want to start a fire. Your match where you light it and then touch it to something, that's your catalyst. That started the fire. That set it all off. That's all it means. Mhm. Remember that enzymes perform strain. They can either break bonds, such as in this example, or make them. When some bonds are broken, others can be formed. So let's think a little bit about enzymes. Enzymes are important even outside of what you do with your own body. Has anybody ever used an enzyme cleaner before? Maybe something like one of these. Because enzymes are good at breaking down materials, they're often included in things that we purchase or use in our day to day life. They're really good for getting rid of smells or getting rid of really stubborn stains because they are good at breaking down the molecules that are part of those smells or stains. The components of those molecules are still there, but you can't smell them anymore because they have a different structure. So enzymes are useful too for things like this. The other thing that's interesting and useful about them is that they are controlled by lots of different things. For example, we've discussed already what denaturing is. Denaturing is the change of structure for a protein. And when a protein structure changes, its ability to perform its function changes as well. Enzymes are proteins, so they are dependent on different temperatures, pHs, and salt conditions. For example, if we were to look at this graph here, where on the bottom we have temperature in Celsius, and on the side here we have the activity of the enzyme, we can see that the best temperature for this particular enzyme is between 30 and 50. Do you know what the human body temperature is in Celsius? What's the typical human body temperature? In Fahrenheit, it would be around 98. 33. Okay, it's a little bit low. It's usually around 37. So normally your body temperature is around here. Did you ever wonder why you get a fever when you get sick? Why do you get a fever, you think? This graph is the reason you get a fever. So, every enzyme has a temperature range at which it works best. So, it's not to release heat after a reaction happens. Your body makes extra heat on purpose. And the reason it makes extra heat on purpose is that sometimes increasing temperature increases the activity of a protein because it increases movement of molecules. So, your basic body temperature is right here on this chart. Peak enzyme activity is higher at 40. This part over here is when you start getting into the 106 range. Danger. This is the point where you would want to go to the hospital if your fever got over here. So, small fevers actually help enzymes work better. But when your body gets too hot, the enzymes stop working. So, your body's better at fighting off infections when it has a slightly elevated body temperature. But it costs a lot of extra energy to do that, which is part of why fevers make you so tired. So, your body doesn't want to do that all the time. And there is a limit. Once you get over that temperature range and you start to get too hot, now the enzymes are going to denature. Once that happens, they will stop working entirely. This is why when your body temperature is over 106, the primary thing a doctor will try and do is get your temperature down. This is also why if your temperature is not that high, under 102, you really shouldn't lower it. Having a slight temperature up to about 102 is actually good for you and will help you recover faster. So enzymes have temperatures at which they work the best, but they also have pHs where they work the best. We talked about how pHs can denature enzymes too. Here are three different enzymes that are found in your digestive system. One of them is in your stomach, and the other two are in parts of your intestines. The bottom down here is pH level. Based on the pH, where 1 is acidic and 11 is basic, which of these do you think is able to work in your stomach? Pepsin. That's right. Pepsin is an incredibly powerful enzyme that's good at tearing apart proteins. This is the primary enzyme that's found in your stomach, and it's able to break down almost everything that you eat. That includes you. So, your stomach has a special lining to protect itself in part from pepsin, but your intestines do not. In order to protect the rest of your body, instead, your intestines change the pH of the environment. As food moves through from the stomach into the intestines, bile and other materials are added to increase the pH to a more neutral and then basic level. So when bile's added, we're going to get over here. And when we get further down here, we're going to get neutralized. This turns pepsin off. So pepsin is still present in your intestines, but it's denatured. The pH is too high, so it isn't able to hurt you. However, if your intestines stop producing bile and aren't able to neutralize the stomach acid, that's when you start getting ulcers, not from the acid itself, but from the active pepsin. So every enzyme has a temperature range at which it works best, but it also has a pH range at which it works best. Outside of these ranges, enzymes denature. Sometimes denaturing is a feature, not a bug. Your body wants to be able to denature some things so that they don't hurt you, but it also wants them to be active where they're supposed to be. Regulating when enzymes are able to do their job is a big part of making sure your body is working as intended. So you should understand that enzymes are sensitive to temperature and pH, that they have a range of pH and temperature at which they work best. You should also understand that it's not all or nothing. Even though they work the best at that temperature, we do still have function of pepsin until it completely denatures around pH 6. It doesn't completely lose its shape right away. It just gets worse at doing its job until eventually it can't anymore. There are other ways to control enzymes too. Aside from denaturing, we can use things called enzyme inhibitors. Remember that in order for an enzyme to do its job, the substrate or reactant needs to be able to sit in the active site. There are two major types of inhibitors. Competitive inhibitors sit inside the active site, preventing the substrate from being able to enter. This is like getting to the seat first on a bus. You snooze, you lose. Non competitive inhibitors change the shape of the active site, which now means that the reactant doesn't fit anymore. So the enzyme isn't able to catalyze the reaction. You should be able to explain ways in which we control how enzymes work. We can control their function by putting them in environments that they do not perform well in, like high temperatures or different pHs. We can also control them by using inhibitors, and you should know the difference between competitive inhibitors and non competitive inhibitors. So, why is this so important? Not only are enzymes important for most of the reactions your body needs to do, they also are what the primary active ingredient is in most drugs, pesticides, and poisons. In fact, some of you probably took an enzyme inhibitor today. Anybody take a painkiller? The most popular painkiller, ibuprofen, is an enzyme inhibitor. Its job is to inhibit the protein that's necessary for processing pain signals. It's not that your body isn't in pain, it's just that your body can't say it's in pain to your brain. So your brain is unaware of the pain your body is currently feeling. Other examples of enzyme inhibitors are things like disulfiram. Disulfiram is an alcohol dehydrogenase inhibitor, which prevents alcohol from breaking down. This means that you can't get drunk. Cyanide is also an enzyme inhibitor. In this case, it inhibits the respiratory chain necessary for making ATP. Fluoride, which is in toothpaste, is also an enzyme inhibitor. This prevents the first step of respiration, which kills bacteria. But it would also kill you if you drank enough of it. Malathion is a popular pesticide, which is also an enzyme inhibitor. Nerve gas is also an enzyme inhibitor. And the antidote for heavy metal poisoning is an enzyme inhibitor too. So understanding how enzymes work, when they work, and where they work can help you control them. And oftentimes we can take advantage of this to treat different diseases or control pests. So hopefully today I've helped you understand a little bit more about how cellular membranes work and things move into and out of cells. You're also a little more familiar about what metabolism actually is and about how it connects to thermodynamics and stored energy. And also a little more about how enzymes function, that they're not reactants or products, that instead they're catalysts that reduce the activation energy for a reaction, and that how we regulate them through temperature, pH, or inhibitors helps us determine when and where they can do the job they're meant to do. That's it for today.